The next tool which is needed for recombinant DNA technology is a cloning vector. So, what is a vector? It is a DNA molecule that can carry a foreign DNA segment or an alien piece of DNA or gene of interest, and it should have the capacity to replicate inside a host cell. So, the most commonly used cloning vectors are plasmids and bacteriophages. This is a plasmid. Autonomously they can replicate autonomously, and they are circular DNA, extra chromosomal DNA. This is a chromosomal DNA. In addition to it, they are self-replicating, autonomously replicating, self-replicating circular DNA, which is seen in prokaryotes. And bacteriophages, they are viruses which attack bacteria. So plasmids are autonomously replicating circular extra chromosomal DNA of a bacteria, a prokaryote. Some plasmids have only 1 to 2 copies per cell. The number of copies in a bacterial cell is 1 to 2, whereas others it can be 15 to 100 copies per cell. And bacteriophages, the peculiarity is that they are having high copy numbers per cell. Okay, and within the bacterial cell, their copy numbers of the genome is greater in the bacterial cell. When the cloning vectors are multiplied in the host, the linked piece of DNA is also multiplied to the numbers equal to the copy number of the vectors. Just now we had told about the copy numbers 1 to 2 or 15 to 100. So based on the copy number, the alien piece of DNA which is linked to it also will be replicated. Suppose a copy number of a plasmid is 100. So 100 copies of the linked DNA will be produced. So the copy number is very important aspect of a cloning vector. So what are the features which are required for cloning into a vector? They are, it should have an origin of replication, it should have a selectable marker, it should have a cloning site, and there are separate vectors for cloning genes in plants and animals. So let us see what is an origin of replication. We know origin of replication or a site is the DNA sequence where the replication begins. So it is a sequence where replication starts. So first requirement for a vector is that it should have an ORI site. This is a modified plasmid vector of E. coli. It is called as PBR322. We can see this is the ORI site. It should have an ORI site. So a piece of DNA which is linked to ORI can replicate within the host cells. In the beginning we had told a piece of DNA cannot replicate as such because it is lacking an ORI site. It will replicate inside the host cell only if it is linked to an ORI site. So a piece of DNA which is linked to the ORI site can replicate within the host cell. It also controls the copy number of the linked DNA. So for getting many copies of the target DNA, it should be cloned to a vector whose origin supports high copy number. So an alien piece of DNA should be linked to a vector which can support greater copy numbers. So it should have an ORI site and the copy numbers should also be greater. Second requirement, it should have a selectable marker. Again, it is a gene. We know plasmid is nothing but a DNA. So it is a sequence of nucleotides. So it is a gene that helps to select transformants and eliminate the non-transformants. In the beginning, in the previous modules, I had told about transformant. When the plasmid or recombinant DNA is received by a host, it gets converted to form a transformant. So only the transformants has to be multiplied and the non-transformants need not be multiplied. So what is the need for a selectable marker? It helps to distinguish the transformants from the non-transformants. So if a piece of DNA is introduced into a host bacteria, this process is called as transformation and that bacteria is called as a transformant. If the transformation does not take place, the bacteria is called as non-transformant. So selectable markers of E. coli, they include genes encoding antibiotic resistance like ampicillin, chloramphenicol, tetramycin, kanamycin, etc. So selectable markers for E. coli are antibiotic genes which code for resistance to ampicillin, chloramphenicol, tetracycline and kanamycin. The normal E. coli cells have no such antibiotic resistant genes. They all have to be modified. Okay, they all have to be modified. Third is the cloning site. To link the alien DNA, the vector needs a single or very few recognition site for restriction enzyme. Isn't it? A restriction enzyme, it is used to cut a vector DNA and also a foreign DNA, that is gene of interest. So it should have only one recognition site or very few recognition sites. If what happens if there are many recognition sites, the DNA restriction enzyme will go and attach to all the recognition site and it will fragment the DNA into many fragments. So that will complicate the gene cloning process. So the number of cloning sites should be one or very few. So cloning site is a site where a foreign DNA has to be linked. Okay, and this is nothing but the recognition site for the restriction enzyme to cut. So ligation of the alien DNA is carried out as a restriction site which is present in one or two antibiotic resistant gene, that is the selectable markers. What is the importance of selectable markers? It helps to distinguish the transformants from the non-transformants. Is it clear? So these selectable marker are nothing but the antibiotic resistant gene which codes for antibiotic resistance. And these antibiotic resistant genes or the selectable markers is actually placed in the recognition sites where where the restriction enzymes cuts and where the ligation of the alien piece of DNA is carried out. Is it clear? So ligation of the alien DNA is carried out at the restriction site. And where are these restriction sites present? It is present in one of the two antibiotic resistant genes. In this case of PBR322, what are the two antibiotic resistant genes present? These pink color regions denotes the selectable markers. They are tetracycline resistance and amphicycline resistance. In the case of tetracycline resistant, it is having the recognition site of BAMH1, the restriction enzyme BAMH1's recognition site is present in the selectable marker which codes for tetracycline resistance. And in the case of amphicycline resistance, the cloning sites or the restriction sites or recognition sites of PDU1, PST1 is also placed. If a foreign DNA is ligated at the BAMH1 site, which is having the selectable marker, that is tetracycline resistance in the vector PBR322 of E. coli. It's a plasmid which has been developed from E. coli, PBR322. And a foreign DNA has been ligated in this region. What happens? This particular plasmid is called as a recombinant plasmid if it has received a gene of interest. And if ligation does not occur, if it has become a failure, then this plasmid is called as a non-recombinant plasmid. Okay, so a plasmid with a piece of DNA is called as a recombinant plasmid, whereas which is lacking a gene of interest is called as a non-recombinant plasmid.